Hello, and thank you for joining our MDICx webinar announcing the release of our framework on developing clinical evidence for regulatory and coverage assessments for in vitro diagnostics. My name is Carolyn Hiller. I'm the program director for our Clinical Diagnostics Initiative here at MDIC. It is a privilege to work with the dedicated volunteers that are involved in our work streams. We'll reserve the end of our webinar for Q&A. Please enter your question in the chat box at the bottom of the screen, and we'll get to those at the end of our webinar. I'd like to introduce our panelists for today's webinar. Jamie Houghton is Senior Director of Regulatory and Clinical Affairs at Sysmex America, Inc. Jamie has over 20 years of industry experience, having worked in clinical laboratory settings as a medical technologist prior to joining industry. She started with Baxter in the blood substitute division and later in Fenwell's transfusion therapies division, where she was responsible for clinical studies of 510K, PMA, and NDA products. She has generated and executed global clinical strategies for various products in the blood collection, processing, and storage areas. Jamie earned a MS in clinical laboratory science from University of Wisconsin-Milwaukee and a BA in biology from Drake University at Des Moines, Iowa. Dr. Louis Jacques is an expert to MDIC and also Chief Clinical Officer and Senior Vice President at ADVI, a healthcare advisory services firm where he is also a partner. He also serves on several institutional boards and advisory panels. Before joining ADVI in 2014, Dr. Jacques was the Director of Coverage and Analysis Group, or CAG, in the Centers for Medicare and Medicaid Services, also known as CMS. He was in that position from 2009 to 2014, where he managed Medicare fee-for-service coverage policy development on technologies as diverse as molecular diagnostic testing, implanted cardiac devices, advanced imaging, chemotherapeutics, wound care, and screening and preventative services. From 2004 and 2009, he was a division director within CAG, focusing on Part B drugs and diagnostic tests. Dr. Timothy Stenzel is the director for FDA's Center for Devices and Radiological Health's Office of In Vitro Diagnostics and Radiological Health. He joined the FDA in July of 2018 and has an extensive background spanning more than 20 years in executive academic and industry leadership, innovation, next generation sequencing and molecular diagnostics, companion diagnostics, global clinical laboratory and manufacturing operations, global research and development, FDA and international regulations, kit and laboratory developed test regulatory authorizations, and commercial launches. He received his MD and PhD in microbiology and immunology, focusing on the molecular biology of DNA replication from Duke University. This was followed by a Duke pathology residency and then a fellowship in genetics and clinical molecular diagnostics. This was followed by a faculty appointment in the Duke University School of Medicine prior to joining industry. As the OIR director, Dr. Stenzel advises CDRH leadership on all regulatory, both pre-market and post-market matters in the in vitro diagnostic, radiological medical device, and radiation emitting products issues that have an impact on center and agency level decisions, policy development, nationwide program execution, as well as short and long-term program goals and objectives. As well, he provides executive leadership and scientific direction to the OIR staff. Now I'd like to provide a brief overview of MDIC for members of the audience that may not be familiar with the organization. MDIC is the first ever public-private partnership created with the sole objective of advancing the regulatory science around the development and assessment of medical devices, which include IVDs. Simply put, MDIC focus is on science, not on policy. MDIC has a robust portfolio of projects focused on regulatory science. You can learn about the different MDIC programs by visiting our website, which is www.mdic.org. As the Program Director for Clinical Diagnostics, we oversee 6 projects within the diagnostic space. The Surrogate Sample Framework that has been completed along with harmonized education on how to use the framework. It's currently with CLSI to be released as EP39 about a year from now. The Clinical Evidence Framework you'll be able to download today. We are also working on projects on using IVD real-world evidence in product development and regulatory submission. We have another project on developing a fingerstick blueprint, which will be study designs for precision and method comparison for fingerstick technology. We also have projects looking at semantic IVD interoperability. And we also are working on somatic reference samples for solid tumors. And now what I'd like to do is give you a high-level overview of the framework. For an IVD diagnostic device, analytical and clinical validity are required for the United States Food and Drug Administration, or FDA, clearance and approval. And clinical utility is required for payer coverage and provider adoption. Many sponsors collect analytical and clinical validity data first to facilitate approval and clearance through the FDA, and then collect their clinical utility data. But this sequential approach to clinical evidence collection can lead to duplicated efforts, changes in the intended use, and as a result, increased FDA regulatory burden, a delay in market entry, or limited coverage. An alternative evidence pathway in which this clinical evidence is collected in parallel can potentially increase the efficiency and reduce the costs of IVD development. The MDIC framework for developing clinical evidence for regulatory coverage and assessments in vitro diagnostics was jointly developed by members from the FDA, the IVD industry, and payer representatives as a tool to help IVD manufacturers develop a strategy to generate clinical evidence for regulatory and reimbursement purposes in parallel. The sections include the introduction, which is an introduction. We're going to define analytical validity, clinical validity, the basic concepts of FDA clearance, and approval of medical devices, including IVDs. Section 2, analytical validity, provides lists of studies frequently used to demonstrate analytical validity, useful terms, test considerations, and requirements related to references are provided. Jamie Houghton will be giving us an overview of this part of the framework. Section 3, the clinical validity, discusses assay types and measures of clinical validation, as well as design considerations for clinical validation based on the intended use of the IVD. Dr. Stenzel will be providing information on this section of the framework. Section 4 includes clinical utility, and this explores the general strategy for developing evidence of clinical utility for payers, represents a clinical utility self-assessment framework that IVD developers can use for planning, and details additional clinical utility considerations by IVD test type. Dr. Louis Jacques will be presenting on this section. And finally, the last section of the framework is on references. This lists the regulatory documents not provided in line with the text and other references cited in this framework. And again, with all of MDIC's frameworks and products that we make available, reading and following the framework that we provided today does not assure FDA approval, nor does it assure payment by payers. All the IVDs are unique, and I encourage you to read the framework and explore the best path possible for you. Now I'd like to turn it over to Jamie Houghton to review the analytical validity section of the framework.
Thank you, Carolyn, and thank you to all the participants responsible for bringing this framework together. I'm happy to be presenting today as a representative of the team. So let's start out with the definition. Analytical validity is the evaluation of how well a device can measure or detect a target analyte or multiple analytes depending on the device. The analytical validity should demonstrate that the device can meet the stated intended use and intended purpose of the device. So how is this accomplished? We go to the next slide. It's accomplished by providing evidence that the results are both accurate and precise, and that there's minimal impact on the results from interfering substances or potential conditions. So we go to the next slide. How do you get started? How do you put together an appropriate analytical study plan? You can start by reviewing this framework, but in addition, reviewing other available sources of information for similar devices. We suggest you take a look at 510K decision summaries for recently cleared devices. These can provide additional details of testing completed by other manufacturers as part of the clearance process. Summaries also provide insight to acceptable testing results. Package inserts can be also a good source of information for testing that's been completed. In addition, be sure to look at any applicable special controls that exist for the type of device that you're studying or developing, or other guidance documents that may exist. The FDA standards database was recently updated and provides a list of standards currently recognized by the agency, as well as sections in specific standards that aren't recognized for certain standards. So there are certain sections for certain standards that are recognized and some that are not. And the FDA standards database actually provides you guidance for that. And then the CLSI guidelines provide the specifics for the different types of analytical testing. So what are the different analytical studies that should be considered? If we go to the next slide, we provide the list of 13 different analytical studies. Those are all included in the framework, and that's a start to the list of analytical studies that you might consider. The framework, as Carolyn said, provides key definitions and considerations for the different types of testing to be completed. It does not provide all the details for each type of test. Instead, we refer you to the CLSI guidelines or other appropriate references for those details. As I mentioned, this is a starting point for considering the different types of analytical testing that may need to be completed for your device. The required testing is dependent upon the type of device, what is being measured, and how the results are going to be used, which are all considerations for the intended use or indications for use for the device. So if we go to the next slide, let's talk a little bit about the intended use statement. So when developing the intended use statement for your device, it's important to define what type of specimens will be tested. For instance, will you be including serum, plasma, urine, or body fluids? If whole blood is going to be tested, what anticoagulants will be included? Will you be including finger sticks or just venipuncture samples, or both? And those are going to be important considerations as you decide what testing you're going to need to do in your analytical testing. Consider where the testing will be completed and who will perform the testing. Will the device be in the main lab, or is it a point of care or CLIA wave device? Will the device be reporting quantitative results or semi-quantitative results, or is it a qualitative assay? And how are those results going to be used? Is it a screening test or a definitive diagnostic test? So we encourage you, as you develop your intended use statement and indication for use statement, that you consider the implications on the analytical testing that will need to be completed in order to demonstrate the device can meet those statements. So the next slide, now we'll go into some of the specific studies that are going to be required. Typically, a comparative study is performed to assess whether the candidate device successfully determines the true value of the analyte. The true value is that value obtained from an FDA-cleared device or a reference method. When selecting a comparative device, it's important to consider the intended use of that device and to ensure it's similar to the device you're developing. Does it include the same specimen types and the same measuring range? If there are differences, you may need to consider a second comparator method, or you may decide to narrow your ranges to match that of the chosen comparator. Either way, you'll need to include samples in your study that cover the entire analytical measuring range for your device. These may be native samples, or you may need to include contrived samples, and we would refer you to the surrogate sample framework as a reference. Next slide. For precision testing, the same sample is tested multiple times under specific conditions. Precision testing includes both repeatability, which is completed over a short period of time with the same operating conditions, and reproducibility, which can be over a longer period of time and includes different operating conditions, including the location, devices, and operators. Sample stability should be taken into account when planning your precision studies, as well as the medical decision levels for quantitative assays, as it's important to characterize the precision at those particular levels as different decisions are being made based on those results. We go to the next slide. For quantitative tests, it's necessary to determine or establish the concentrations at which the test is linear. This can be accomplished by using 7 to 11 samples of known concentrations or known dilutions. Testing of each sample is completed with multiple replicates, depending on the expected imprecision of the assay. The highest concentration tested should be above the upper limit, and the lowest concentration tested should be below the lower limit of the linearity interval. Next. An assessment of the measurement boundaries of the assay is required. This typically requires determining the lowest level of detection capability. There are multiple methods for determining the LOB, LOD, and LOQ, and those are dependent on the analysis system, the test methodology, and the analyte being measured. It's also considered important to consider the fact that the LOB, LOD, and LOQ may need to be established for each specimen type. We go to the next slide. The susceptibility of the device to accurately quantify the results in the presence of potentially interfering substances must also be characterized. Both endogenous and exogenous sources of interfering substances should be considered and applicable testing completed. Labeling for existing similar devices typically provide a list of interferences evaluated, and those interferences can be a start for your testing. Go to the next. And then it's also important to determine the normal range or reference interval for the analyte using the candidate device. You should consider the impact that age, gender, race, and ethnicity may have on the reference interval, and separate reference intervals may be appropriate. So now that you've defined your analytical study plan, you can taking into account the intended use statement. You can put the details of your plan together into a pre-submission document and submit that to the agency to obtain feedback on your proposed testing. And that summarizes a little bit of the analytical section of the framework. And now I'll turn it over to Dr. Tim Stenzel for the clinical validity review.
Thank you very much. Pleasure to be part of this webinar today. So yes, so in the next slide, already covered, let's see, okay, is analytical validation, and I'm going to talk about clinical validation. So clinical validation is a patient test result associated, the question is a patient test result associated with the expected clinical presentation of this patient? And specifically, the process through which one shows that test results are clinically meaningful. Next slide. So, depending on how well the analytes are known, different levels or different amounts of evidence may be required. Where there's a lot of experience with an analyte, or in other words, an old analyte, where clinical validity and perhaps even clinical utility is well established. Largely analytical validation may be all that's required. This is a case-by-case determination. An example might be calcium in serum. On the other hand, there may be totally new novel analytes or combinations of analytes that are a new combination where a clinical validation study may be needed. This might be, for example, a new biomarker for detection of colon cancer. And then the last major category could be an older analyte that has a new purpose, a new or changed intended use. And, to be able to show that that's clinically valid, a study may be needed. An example of this could be the example of CRP, where originally it was not developed or discovered as a risk factor for cardiac disease, but later was determined that that would be a good use for it. And then in that case, studies to prove that for that new intended use may be required. Next slide, please. So some of this has been mentioned before in this webinar, but it's important to look at what your intended use or indications for use are going to be. What does the device measure or detect? How does it report this? Is it qualitative or quantitative? What are the actual specimen types? What's the target intended population? Age, sex, whatever. Who are the intended users of this technology? Is it an untrained user, a potential point of care, over the counter, or home use device, or is it something that only can be performed, say, in a high complexity lab? And then, what is the target condition, the disease, and what are the contexts? So, what is the test providing? Is it providing, and we'll go into some of this, I think, in a subsequent slide, but, is it for diagnosis, screening, or monitoring, for examples? Next slide, please. So actually, this is a really good thing to do, is to write out the intended use or indications for use as you're developing your plan. It really focuses the developer on what's really important, what they want to claim, what they want the test to do. And all of this will be key in evaluating what appropriate clinical studies may be needed for such claims. And so just to walk through this specific situation, so many of the elements that I've discussed prior to this slide are equivalent here. So of course the test name, in this case it's a qualitative rather than a quantitative test. It's a single laboratory, so it's not a distributed kit. It uses a gene expression profile. Those are the analyte, that's the analyte or the analytes. The sample types are clearly established here. In this case, it's fresh frozen breast cancer tissue, so that's different than FFPE, and obviously if you're doing gene expression profiling, that specimen type, whether it's frozen or FFPE, can be critical to how well the test performs, and it's important to demonstrate on the intended use tissue. You know, what's the target population? Of course, breast cancer patients. Age is included, stage is included, tumor size, lymph node status. And then, you know, who is going to receive these results? In this case, it's physicians. And what's the intended purpose? It's, you know, indication for use. It's a prognostic marker. So it's not diagnostic. It's not for screening. It's not predictive. It's not for, you know, determination of particular therapy, whether that'll be effective or not. In this case, along with, you know, other information from the patient. So that's a real good example to explain all these things and why, and potentially why they're so important. Next slide, please. So your design, if clinical validation is required, depends on what we just discussed, intended use and indications for use, and then, you know, what your intended performance characteristics are. So what do you believe the clinical sensitivity needs to be? What do you believe the clinical specificity needs to be? And then, given the incidence of the disease in a population, what do you think the intended predictive values are required for your test to be successful? And, you know, in coordination with the agency, you know, what does the agency think the targets need to be? Then you design your study around making sure that you meet those endpoints. You power it using statistical means to make sure you have enough patients included so that it's not discussed here, but so that your confidence intervals are also acceptable to the agency. So, you know, how novel is the technology? Is it something brand new? Is it the first time the agency is seeing? Well, that might, you know, then of course be a de novo, or it may be a PMA, depending on the ultimate risk level of the device. But you know, if something's brand new, then some additional factors maybe need to incorporate it into the study. And then, of course, what is the availability, if it's a brand new technology, of appropriate method to establish what truth is? And that may not always be just with novel technologies, but it could be for other types of examples as well. Next slide, please. So previously we briefly discussed, you know, what is a testing context for IVDs. So we talked about diagnosis, screening, and monitoring. Well, there are other categories too: risk assessment, prognosis, therapeutic response, prediction, say for a companion diagnostic. And each of these contexts would perhaps describe or require a different sort of study design. So screening, you know, is a pretty big, you know, study to be able to show that in a general population that where patients are asymptomatic and are average risk. Maybe, you know, perhaps one study design versus a subpopulation that it's already at a higher risk, where the incidence is going to be higher, and that population perhaps a more straightforward, a smaller study design would suffice. For prognosis, you're obviously going to need to know what the outcome of that population was that you're studying. You know, did the patients do well or not do well based on the test results? Was it truly a prognostic assessment? And then monitoring, you know, how frequently do you monitor? Do you establish a baseline before you monitor? Those are all really important considerations when you design a trial or study for showing clinical validity of those different types of situations. Next slide, please. And the last few slides that I'm going to go through primarily are going to talk about samples for use in clinical validation studies. And and when and where they can be used, and what considerations you have concerning should consider for them. So, and obviously the highest level of evidence would be on prospectively collected or obtained samples. If done in the right way with the right inclusion and exclusion criteria, you'll have a very strong study. You will have largely eliminated bias from the study design. However, that is typically a more challenging study to do for various reasons, and may not be required, and many times isn't required. But depending on what your intended use is, it could be required to have this sort of study design. The next kind of samples are archive samples. I'll talk a little bit more detail about that, but these are already previously collected samples, and there may be different ways of doing that. And there are considerations when using those. And then for particularly difficult samples to obtain, the agency may allow surrogate samples to be used, perhaps to augment rather than replace a clinical study. And then, it's important to talk to the agency about those. But I'll go into a little more detail with all of these. Next slide, please. So prospectively obtained samples usually are collected under a pre-specified protocol, that describes inclusion-exclusion criteria, the sample type, what your study design is. These are very important documents to get right and be reviewed before starting your clinical trial. So, are the sample subjects from the target population? And, then it would measure the clinical performance in this setting, and largely as described before, in an unbiased manner. Now, there are two different timing considerations that can be made. You can do a prospectively obtained sample study but delay testing. So, and that, falls under this category. This may be a very more efficient way to do the testing on the samples, because you can batch these samples. That can lower the cost of the study, and then it in various ways, and is a totally acceptable way to do this. Or there may be situations where testing is needed immediately. That might be for, say, a companion diagnostic, where you need the information to be able to make the therapy selection. And in that case, doing immediate testing may be important. However, there may be situations where the testing in the clinical trial was perhaps done with another device. It might have been an LDT or something. And now it's important to get the drug approved and the device approved to do a bridging study. And we didn't go into detail in these slides, but again, that would fall under this category if those samples were obtained prospectively and they were, in this case, stored. There are bias situations where you want to avoid, such as you want to, not be able to link back the results of the device testing with the prior testing that was done. So those considerations can all be worked out. Okay, next slide. So moving over to the use of archived samples. This could be with or without a pre-specified protocol. It might have been a protocol for a different study. And so they may have been collected in a manner that's totally acceptable for this new use, this new study. However, there are potential challenges here. Are there non-represented populations within the study? That is, if the only samples that are left over in this case are from, say, large tumors, because there was plenty of sample, but the small tumors, all the material was used up, and you don't, so they're not represented in an unbiased way, and that, those are important considerations to make in finalizing the clinical trial design and how you approach this. Also, you may have samples that are more challenging than others. They may be two, say, tumors of equal size, but one's a lot more challenging than the other, and in due course of testing that more challenging tumor, you may use up all the sample as well, and then therefore you may have an underrepresentation of the more challenging samples. If you've selected those samples in some way, positive selection such as using another test, that could introduce bias that will need to be addressed. And then the other thing with archived samples is there may be samples, especially if they come from different sources, that are stored differently. You may have stability issues, and so it may not be an unbiased population. So storage conditions of archived samples are very important, and to address any potential risks. One of those might be that the older samples are less stable than the newer samples. Next slide, please. So last couple of slides, last two slides. Continuing on with archived samples. The measurement of clinical performance, depending on the study's design, could be biased, and the study design, for it to be acceptable to the agency, must address these questions in an appropriate manner. And then, that's probably best done interactively with the agency. So next slide. And finally, surrogate samples. So, we do allow the use of surrogate samples in certain situations. For example, if you need to augment the number of samples that are near the limit of detection or near a cutoff, if the unbiased population just didn't have enough samples around this, that could be a situation. Or you may have very rare genotypes that are not well represented, and to be able to represent them more fully in the study, a surrogate sample could be designed. And obviously it makes sense to contact our office before the use of archived and/or surrogate samples to properly address any concerns we may have about the study design. And with that, it concludes my portion of this webinar, and I turn it over to Louis. Thank you very much.
Thank you. And I think the first two speakers have wonderfully illustrated why a payer would much prefer that you go through FDA rather than presenting them with an LDT or a laboratory developed test, because all the details and the nuances around analytic and clinical validity are much more readily dealt with by the payer if an entity like FDA has given it its essential, seal of approval. Otherwise, a payer that is more predisposed to be looking at drug data or medical device data from an implanted sort of device, or a procedural outcome from a surgical procedure, has to literally dive into the weeds of analytic and clinical validity in a very technical way. And that is not the way that the typical payer is staffed internally. So if you could imagine the typical payer having to literally go through all the factors that have been talked about over the last 30 minutes, you can probably appreciate why payers in general do not welcome conversations about LDTs. They would much rather be able to look at an FDA-approved label and say, okay, we know what you claim to do, and we know that within the confines of the label, you reliably do certain things. Now we'll have a discussion as to whether those things that you do are essentially within the scope of the insurance benefit and if they add clinical utility. So let's talk about clinical utility for a bit. Next slide, please. Medicare and payers did not invent clinical utility. This is a discussion that has been going on for quite a long period of time. Some of you may remember this particular wheel came out of the EGAP and ACE initiatives that were undertaken by the Centers for Disease Control and Prevention and others in the early part of the 2000s. CMS was represented on the Secretary's Advisory Committee on Genomics, Health, and Society. So CMS had a seat at this particular table, but CMS was not driving this conversation. This was a conversation that would happen whether or not CMS existed or not. But to that extent, it was certainly consistent with the way CMS interprets its own legal authorities. So CMS enthusiastically embraced it. Next slide, please. Clinical utility is the coverage standard. It is not clinical validity. It is not analytic validity. It is clinical utility. Medicare, like other payers, defines clinical utility as being demonstrated by evidence of improved beneficiary health outcomes when the test is incorporated into the management of that patient's condition. You're not required to cure the patient. There can be a test-based avoidance of futile, burdensome measures, which could be considered a positive utility, and I have some examples of that later. There can be a direct or an indirect chain of evidence. In a direct chain of evidence, you've actually done a clinical trial to directly assess the impact of the test incorporation into disease management and patient outcomes. Indirect evidence occurs when you can assemble a logical chain of evidence leveraging what may already be known, for example, in the therapeutic side. So if you look, for example, at cardiology or at oncology, the therapeutic sides of oncology and cardiology are reasonably well developed compared to some other clinical spaces. So you may, in fact, if a lot is already known about the downstream impacts of a particular clinical status for a patient, you may not have to reinvent the wheel. And a payer may say, we know what happens downstream from here, so as long as you get us to here, we are okay. Someone decided to sue the Secretary of Health and Human Services a few years ago. At the time, it was Sylvia Matthews Burwell. So the federal court in the 4th District was actually asked, is Medicare's requirement that the impact of a test on health outcomes in Medicare beneficiaries needs to be essentially articulated and supported by evidence, is that a reasonable position for the agency? Because the plaintiffs were essentially saying, you know, gee, that's unfair. We're a test. All we do is furnish information. Information's a good thing, and it's unfair for the secretary to consider whether using this information would actually have an impact on health outcomes. Well, the secretary has a pretty good track record of winning on these sorts of things. And the court affirmed that Medicare's interpretation of its own authority was consistent with a good reading of the statute itself. So it is clinical utility. Clinical utility is not merely changes in physician management. Payers have no interest in rewarding Brownian motion by physicians. It has to be changes in management. In other words, what happens to the patient that wouldn't have happened without the test? If the test simply leads the patient down the same management path that they would have been on otherwise, there really is no clinical utility for the test. So as an example of not reinventing the paradigm, clinical chemistry is relatively straightforward, as the previous speaker noted. And it's well established that if a patient has excessively low or excessively high potassium levels, which is K, if you're not into clinical chemistry, that the patient will develop certain cardiac dysrhythmias, which may in fact lead to morbidity or mortality. So if you were to devise a new way of measuring serum potassium by something that vaguely resembles current paradigms, in other words, not a PET scan for serum potassium, but if you were to come up with a new assay for potassium that reported the outcomes in milliequivalents per liter or something that a physician was used to dealing with, the typical payer, number one, wouldn't notice, and number two, wouldn't care that you had done it, because that particular paradigm is pretty mature. I can't imagine any payer saying, well, you need to do a cardiovascular mortality trial to see if we can get the same benefits from treating hypo or hyperkalemia if it's diagnosed via your assay versus something else. Of course, those aren't the kind of tests that come to the attention of payers. It's all about novel biomarkers in cancer and a few other conditions. Next slide, please. So this is the kind of combination lock that most of us had in college. And, you basically, 2 turns in one direction, 1 in the other, and a half turn in the other. And it's usually just 3 numbers. So from a patient's point of view, if you only need 3 numbers to open the lock, why would it be reasonable or necessary to obtain 4 or 5 numbers? If anything, that excess information simply increases the likelihood that you will enter things in the wrong order, because now you have, frankly, more options in which to enter the numbers. Next slide, please. So this is the case itself, Court versus Burwell. It's also been called Smith versus Burwell. But essentially, as the court has concluded above, defendants, defendant in this case is the secretary, defendants' consideration of health outcomes and disease management was permissible under the Medicare Act itself. And the link is there on the bottom if anyone wants to look at it. This particular case was about the national coverage determination on positron emission tomography for the identification of beta amyloid in the brain in patients who were suspected of having essentially dementing conditions, specifically Alzheimer's disease. Next slide, please. So why would CMS embrace clinical utility? As I mentioned at the outset, CMS did not invent the paradigm, but it's been well articulated. It's been subject to a very large amount of public input and public comment through the activities of various other agencies. And CMS's advisory panel, the MedCAC, the Medicare Evidence Development and Coverage Advisory Committee, has endorsed it many, many times. It's also consistent with prior national coverage determination language, which speaks to looking for evidence of improved health outcomes. CMS has historically referenced the landmark paper by Fryback and Thornbury, who are a couple of radiologists, but specifically talking about clinical utility and its impact on patient health outcomes. And it's compatible with existing regulations. That's Title 42 of the Code of Federal Regulations, Section 41032A, which talks about the use of a test by the beneficiary's treating physician in the management of that patient's specific medical condition. Now, meeting that reg is not in and of itself sufficient for coverage. It is simply a baseline that if you can't meet that, you are fundamentally not eligible for Medicare coverage in the first place. Next slide, please. So when Medicare looks at desirable evidence, and this is an animated slide, so if you sort of click it 3 more times, 3 arrows should appear on the left side. What payers are often confronted with, and I don't mean to use the word confronted in a confrontational way, but mentally you are sort of confronted by a dilemma. Payers are often presented with what I would call reasonable analytic validity evidence and immature or incomplete clinical validity evidence. For many of the reasons addressed by the previous speaker, there may be all sorts of questions about, biases from the use of archived samples, et cetera, et cetera, et cetera. Are there any essentially non-generalizability challenges posed by the way the previous studies were done. But in general, what you have is you're presented with a dossier that says, well, here's our analytic validity. Here's our test performance under a variety of laboratory conditions. And here's our clinical validity based on multiple samples from an archived cancer trial that had probably, 400 people in it. We managed to get 212 samples. And gee, this is what we got, and we kind of resampled it a few times or used half of it for validation and the other half for testing or for generating the paradigm in the first place. And from a payer, it's like, well, okay, that's fascinatingly interesting, but it's not really persuasive. What we really want is the green arrow, which is evidence of patient health outcomes. So, does the patient experience the cognitive or functional decline that is abated or potentially arrested if the test indicates that this patient should be taking a therapy other than the one that they're taking now? Can you potentially delay institutionalization? And this is in the context of a cognitive disease. Can you avoid certain adverse events from certain medications because the test says that the patient wouldn't respond to those medications anyway? Payers in general don't get to societal outcomes because essentially the aliquot or the unit of analysis for a payer is essentially the patient or the beneficiary in the case of Medicare. On occasion, we get something in the middle, which is, we have, a lab comes in and says, well, this is our clinical validity. We think it's pretty good. And we have 3 KOLs who insist that this test is absolutely valuable for them in their practice. Well, okay, that's interesting. How representative are these KOLs who happen to be on your payroll or your research support group or your speakers bureau? How representative are these KOLs more broadly about physicians in this specialty? Are there any physician specialty guidelines that specifically recommend the use of this test, i.e. a physician should use this test, as opposed to a physician might use this test or a physician may consider using this test? Which I used to joke meant that if the physician rolled off the right side of the bed, they should use it. If they rolled off the left side of the bed, they shouldn't. Some guidelines aren't particularly helpful, but because what the payer wants to know is there adequate evidence that a physician who is acting in a manner consistent with the evidence and presumably has the patient's outcome interest, i.e. they want to make the patient better in the end, is there evidence that the physician community has in fact accepted this paradigm and that this test itself is useful in that paradigm? So physicians may, for example, in an oncology setting, have significant agreement that a particular biomarker has clinical utility. So then the question is, well, how well does this test, especially if it's an LDT, precisely, repeatably, accurately assess that biomarker in a clinically useful range of values? And are physicians sufficiently convinced that that happens, that they actually do make treatment decisions based on this particular biomarker? So from a payer, depending on what kind of evidence comes in, you have to sort of attack the issue from a somewhat different perspective, because you may have certain givens that are fairly, well established, or you may almost be starting from scratch. Next slide, please. So here's the rebuttable presumption of clinical utility. In this particular context was in a prognostic test for patients with prostate cancer. This particular one was to try to guide management in patients who had already undergone radical prostatectomy. And, is there some value to aggressive treatment further on? Payers recognize that it takes a long time to do a prostate cancer trial because it's a very slow growing indolent condition, and it simply takes a long time to accumulate enough events to be able to have a comparison of event rates between two arms. There is also a general understanding among clinicians payers and sometimes also patients that most men with prostate cancer are overtreated, and that treating 100% of men to potentially get a benefit in 4, 5, maybe 10% of men results in a lot of men undergoing invasive therapies that adversely impact quality of life and may in fact impact negatively other morbidities as well. And isn't there a better way to do this? So what some payers have done, and this is Noridian and Palmetto, both Medicare contractors, what they said in this case is we're willing to assemble this logical chain of evidence and say, based on what we know happens on the therapeutic side of prostate cancer, and based on what we know based on the natural progression of this disease and the difficulty in figuring out who's high risk for progression and who's not, we will essentially extend a rebuttable presumption of clinical utility. So in other words, we will go ahead and cover the test until and unless subsequent evidence indicates that this presumption should in fact be rebutted. So they weren't sitting there drawing a line in the sand saying, well, you better start enrolling a prospective clinical trial now, and by the time you're done, it'll probably be 15 or 20 years. They were willing to say there's enough known in the bookends before and after this test that we are persuaded, at least for the time being, based on the available evidence, that there is clinical utility here. We're not 100%, 1,000% confident, but we're confident enough, and we're going to be reasonable about it. Next slide, please. Here's another one, and this is a national decision on positron emission tomography, specifically in the context of cervical cancer. What is known is that if a woman has a positive supraclavicular node, meaning above the clavicle or above the collarbone, that curative management is unrealistic. Because if curative management were a realistic option, these women would have extensive pelvic surgery. Now, to subject someone to extensive pelvic surgery, if there is no reasonable likelihood that she will benefit from it, seems barbaric at best and maybe cruel at worst. So even though the incorporation of the PET scan does not improve her cancer-specific outcomes, I mean, the cervical cancer is going to do what it's going to do, subject to the availability of therapies. At least she can avoid having surgery, which imposes near-term adverse events, whether it's hospitalization, infection, incontinence, pain, anything else. So when you remove a bad health outcome, you essentially overall end up with net improved health outcomes. Next slide, please. This is Fryback and Thornbury, and there was another citation from Mall and colleagues, and these are all in national coverage determinations. This is literally boilerplate. If you ever see a national coverage decision analysis on a diagnostic test and you look up Fryback and Thornbury, you will find these. So as they note here, and as I highlighted in yellow, it's the middle one, consequently, the practical value of a diagnostic test can only be assessed by taking into account subsequent health outcomes. If a particular diagnostic result can be shown to change patient management, which is one step, but in and of itself not sufficient, and other evidence has demonstrated that those patient management changes improve health outcomes, then those separate sources of evidence may be sufficient to demonstrate a positive health outcome from the diagnostic test. And that is essentially the principle that was articulated in the particular in both of the prior examples, both in prostate cancer and in cervical cancer. This is probably much simpler to do in cancer. As I mentioned earlier, cancer care in this country is reasonably guided by guidelines. Now, not every guideline is perfect in every cancer, but compared to a lot of other things, cancer therapy is fairly well articulated. And this is an important and, most cancer therapies are lethal enough or otherwise harmful enough that if you gave them to your neighbor, you would be arrested for attempted murder. Okay? So absent having a medical license in a patient who has a condition that makes the risk worthwhile, these are not things that one would simply take, for grins. So when you can avoid exposing a patient to something that is potentially that harmful, it's almost a slam dunk of low-hanging fruit to say that you're improving the outcome by not poisoning the patient or by not imposing a surgical procedure on a particular patient. That paradigm is not always available in other clinical spaces where the various therapeutic options may frankly be quite benign. They might even in fact be over the counter in some cases. Next slide, please. So I'm going to punt all of this back to our moderator, Carolyn.
All right. Thank you very much to Jamie, Tim, and Louis for giving an overview of the framework. What I'm going to do before we go to our questions is I'm going to ask each section lead, what do they think the key takeaway is from their section? I'll start with Jamie with analytical validity.
Well, thanks, Carolyn. I think that the key takeaway is just that we've outlined 13 different analytical tests to consider. That may not be all the tests that you need to complete as far as your analytical section for your submission, but it will get you thinking about the different considerations when you put your analytical testing together. And then to look at, as I mentioned, the other sources of information so that you have the most robust study design possible.
Thank you. Tim, would you like to tell us what you think are the key takeaway from the clinical validity section?
Sure, yeah. So how you design your study, and even if a study is required, depends on the analyte and the intended use, and that defines what may be needed. And of course, conversations with the agency are great, always. They're not required, but welcome prior to doing your work. And then the other thing is that the agency is flexible in the approach of using something other than prospectively collected samples, so depending on the situation. So if that's something you want to entertain, whether it's archived samples or it's surrogate samples, we invite you to come have a chat with us. Thank you.
Great. And Louis, what's the
Sure.
key takeaway from the clinical utility?
My suggestion would be that before you even get started developing a test, that you flow chart the typical experience of a patient who has the condition in which you're trying to work. How does the patient present? What are the important clinical questions that arise? What are the therapeutic options? And what is the usual outcome from a patient that goes down any of those therapeutic paths? Then figure out where your test fits. Is your test early in this history? Is this later in the history? Is it at a decision point between two tests, or between two therapies rather? And then ask yourself, well, depending on the test result, what is the physician going to do differently? And I think if you can start from there, you're in a better position to be able to articulate your evidence. Because too often what I see, or what it appears to me it seems, is that someone said, hey, look, we have a sequencer and we got to find something to sequence. And hey, we have all these samples. So let's go fishing around, see what we find, and then figure out if that's useful for anything. And I'm going to suggest that that's a very dysfunctional way of going about something if you want to get paid. It may be fine for other things, for scientific exploration of topics, which certainly has its own value. But if you're looking to go to a payer and say, I want you to pay me to do this, I would recommend sort of flow charting the whole clinical pathway first.
Great. Thank you, Louis. Now, we do have a question here from one of the participants, and I'll leave it up to the panelists to decide who wants to answer this. And the question is, what type of clinical evidence is typically required for devices that are used for sample preparation, but do not generate a test result? For example, a device used to enrich the analyte of interest prior to downstream testing.
This is Tim. So that specific situation really wasn't covered in this webinar. So, but there are a number of considerations that would be used. Since it is kind of off topic, we do have our pre-sub or Q-sub program that has no user fee, so it's a free service just to reach out to us and engage us if you have specific questions about something you're considering.
Great, thank you very much.
If I can, yeah, can I chime in on that as well? Because I think from the payer point of view, one of the questions that is going to come up is what exactly is in the test itself, which may involve what does the coding in the long description say that the test is, because a test is going to be paid based on essentially the amount of work it takes to do it. So I think if someone goes to FDA and says, hey, we have this other thing that prepares the sample or that concentrates, circulating tumor cells or whatever else, and then there's an assay performed downstream, the payer may still have some interest in, well, unless you're planning on doing all the rest of that prep work for free, we want some evidence that you're actually, using something that is appropriate for clinical use. So just sort of keep that in mind. In general, from a payer perspective, not that I want to give FDA more work to do, but essentially from a payer perspective, the more completely your test has gone through FDA, as opposed to saying, well, we'll send this bit to FDA, but not this other bit. I think the more comfort a payer is going to have that you're, sort of staying on the highway.
Great. Thank you. And I don't see that we have any other questions. I know we've gone over our time. I want to thank everybody for staying with us. A special thank you to the volunteers that helped create this framework and today's speakers. If you have questions about how you can engage in MDIC, please feel free to reach out to me. You can reach me at my email, which is chiller@mdic.org. You can download the framework on our website, and I have the URL listed here on the slide. If you don't capture that, go ahead and Google MDIC IVD clinical evidence, and it should be a top hit. Thank you very much, and I wish all of you a very good day. Bye-bye.